Hello, everyone. Good morning and happy Friday, and welcome to another installment of MBF Bioscience's new short-form webinar series called TJIF, or TJ's Information Fridays. I apologize for the delay in getting this installment out to you guys, but nonetheless, I'm happy to have you all joining me. If you've attended some of our previous webinars, you might remember me, but if this is your first time, I'd like to introduce myself. My name is Tetreault, and I work here at MBF Bioscience in the MBF Labs group. And I do a wide range of things from quality assurance work to demonstrations online or evaluations of customer data to see how the program might work for them, and a little bit of everything in between. And so I'm excited to kind of keep the ball rolling with this webinar series and to be able to introduce a short form way of getting our tools across in a way that might not necessarily come across in a regular demonstration. So again, thank you for joining. As the title says, this week I'll be focusing on the new Subvolume mode that we've implemented in our latest version of Neurolucida 360. And I'll be going over ways not only of just how to use it, but potentially ways of how you and your team can optimize data generation within the images that you guys have. Although this is a shortened webinar, the overall success does depend on your curiosity and the questions that you ask. So I'll quickly go over how that can be done. If you develop a question as the webinar continues on, all you'll need to do is find this red or orange arrow, click it to extend that webinar panel, and then you need to find this questions box, type it in, and hit enter, and that will be sent directly to me. I'll do my best to address the questions as they come in, but please forgive me if I don't get to them right away. This is just my second solo webinar, so I'm still trying to figure out and navigate through the multiple screens and the multiple programs that I have running. I have set aside a few minutes at the end of this webinar to answer any questions that are still remaining. So if I don't get to them right away, hopefully I can get to them by the end of today's webinar. Lastly, the webinar is being recorded and will be available to view at a later day and time. And a link will be sent to the email that was provided. So let's jump into it. On the screen now, you should be able to see, let me just make sure that you guys are able to, perfect. You should be able to see a nice large image. So this is actually an image that we've used in the past to demonstrate our large volume reconstruction mode. And what's nice about this is that we no longer have that mode. We've replaced it with our Subvolume tool. The first thing that I want to get across is that this Subvolume tool is not just for the Neurolucida 360 Studio users. This is for anybody that has Neurolucida 360. So that's a major difference between the old large volume reconstruction mode and this new Subvolume tool. So this is applicable to anybody who has large data or very dense data and anywhere in between. We can really use this tool to kind of dig into the research that you're looking to do. So right away, what I'm going to do is I'll click on this Subvolume tool, and you'll notice that we have a nice cubic area generated right in the center of our screen. I can click and drag to move that Subvolume area around. I can also just click on my image to have the Subvolume center on the area that we're interested in. So here, I'll click on the soma, and then I'm going to zoom in just so I can make this the proper size for my liking at this point in time. There's a couple different ways that we can change the size of the Subvolume. The first is by holding down the Shift key and using your mouse wheel to push forward or pull back to change. So this is a nice way to systematically change the entire volume in one fell swoop. If, however, you would like to have a little bit more of a manual sense of how large your region is going to be, we do have these 6 different spheres on each side of our cube that will represent that side of the cube. As you click and drag, you'll notice that that portion or side is being pulled or pushed depending on the way that you move that sphere. So this is another way that we can manually change the region that we want to work within. So I've made mine a little bit more oblong, more rectangular than cubic. And that's just the way that I've generated it here. It doesn't necessarily matter. It's more about how you need to regionalize your data. From here, I'll just click this subvolume button, view subvolume. And in just a quick second, you'll notice that we've zoomed in and the resolution has become the high-powered resolution, the full resolution of the image. This is really helpful because if I return back to the full volume quickly, you'll notice that because of the way that the imaging engine works, we're loading the whole image in at once, but it doesn't necessarily mean that it's all going to be at the highest resolution to start. Clicking that subvolume button will bring that full resolution to the forefront, which is very nice. Another helpful tool here is this Save Subvolume button. In doing this, I can name the subvolume or the region of interest that we're working in, click OK, and it will be pushed to our saved locations area. This is helpful for your current session as you continue to work and build data, rather than having to return to the full volume and search for a specific area that you might have already been. You can just click load, this blue arrow, and the program will bring you directly back to the area that you've already started. And this can house multiple saved locations. So this is going to be helpful as you continue to build data, as I said, during your current session. From here, I can just start generating data as we normally would within any image that we've brought into Neurolucida 360. So from here, I'll do a soma detection, and I'll do a manual detection to click and detect. And then I'm going to slowly shift my image. I'm using my hands, I apologize. I'm going to shift it to the right. From here, I'll go to tree, and I'll do the user guided mode to start. Pardon me. And again, this is just like working with any other image that we've had in Neurolucida 360 before. All I need to do is click and trace just as I normally would. And as I get to the edge of my boundary of my ROI or my subvolume, you'll notice that the program generates and moves. It will generate a new subvolume for me to work within based on the direction that I'm moving within the current subvolume. So because I'm working toward the left, the program is going to systematically build and shift that ROI to the left as well. I'm going to continue to trace just a little bit more. And now we can see a much vaster shift within the program, or the ROI rather, the subvolume. Once again, I'll continue and load the next region. And the nice thing here, I'm going to right click and end the tracing. So it stopped it right when I entered. And so if I zoom out, I can see that the soma is still reconstructed. I can see the branches that have already been reconstructed in this image. And now I can see the subvolume that I'm working in currently. There's no necessary reason to continue tracing the same path if there are other, let's say, processes of interest that you're looking at. So I can go ahead and click up here on this top branch, and I can continue to trace as if this were my original, and I can work this way. As I get to the edge, once again, it will generate that new subvolume area, pardon me, and I can continue to work this way. Excuse me again, I apologize, everyone. If I click this home icon to reset the orientation, what it will do is it will not push out to the entire image, but what it will do is it will keep the boundary of the original subvolume around that soma that we had, and it will use that as the boundary because that's where the data generation had started. If I go back to this subvolume mode, you'll notice that we can see again our subvolume square, but when I click view full volume, it will once again pan out to the original volume of the entire image. Let me shrink that back down. I had already mentioned the load. You can also delete these areas if once you've returned, if you feel like you no longer need to work in that region, you can always delete these as well. So that's just a very basic introduction of the subvolume mode. There are a couple of nice things to note with these large images. If I click back to view full volume, you'll notice that as we zoom in, there's an axon that follows all the way out. And as we get closer or further away from that cell body, it's becoming much less apparent to the eye. We can click this view subvolume button. And again, as I mentioned previously, we're looking at that full resolution of that axon. So we're able to trace throughout this. We can use the user-guided mode, the smart manual mode, or the automatic mode, any tracing algorithm or mode that we'd like. Once we enter the subvolume mode, it's going to be treated as if it were just a normal image stack. So this is nice for images that are very sparse. But another thing that can be helpful is when you're working with really dense images. This subvolume mode will allow you to create a nice region of interest and allow you to kind of push your data and figure out how best to optimize your tracings. So I'm going to quickly shift over to a different image set or image stack. And so we can see here that this is very different from the original image that I just had on the screen. Excuse me. Here, obviously the image is much smaller, but the data is a lot more dense. You can also see that the saturation in the somas here is very prominent, and the branches that are coming off of them below, the staining is also very prominent. But if we look towards the top, there are some somas and some scattered branches above, and the signal seems to get a little bit more sparse. So with this subvolume tool, what we can do is we can kind of regionalize where we want our automatic detections to go. Let's see. Sorry, I got sidetracked for a second. So what we can do is I'll click the subvolume mode once again. I'm going to move my boundary box up to the top, and I can drag and create a nice region of interest or subvolume above the somas that are saturated. And again, I'm going to do this in all three axes. So now we have a nice little region of interest that might require different settings. So I'm going to view the subvolume, and we can see here we have our full resolution once again. I'm going to go and do another manual detection of the soma. The only reason I'm doing the manual detection here is because of the image size. I've done some testing with this image while running the GoToWebinar program, and it would take almost the entire webinar or a good portion of it to detect the somas in this image. So rather than wasting time and making you guys watch and sit and see a screen that's just loading, I'm going to do a manual detection. And for demonstrative purposes, we'll say that all of the somas have been detected. I'm going to go to tree. And the first thing that I'm going to do is in this user guided mode, there's an option for a typical process width. What this is doing is as the image is loaded, the program is getting a rough calculation of the overall diameter or width of the processes, and it wants to average that out. So that way, when we go to do our automatic detections, it's going to be looking for processes about that width. That does not mean that we're ignoring smaller or larger diameters. It's just giving a good basis for the algorithm for these seed placements to start. So the reason why I'm saying this is because in the overall image, the branches below have a lot larger of a diameter than the scattered branches and the signal up top. So what I'm going to do is I'm actually going to lower this threshold to 10 microns. And so now we should be able to detect some of these finer processes with a bit more ease and a bit more accuracy, rather than having to comb through the data that's going to be produced afterward and go through the editing process there. So this is just a quick jump to hopefully beat the editing process and save us some time. I could click trace, but what I want to show is that we're going to use two different settings for the same image and show how powerful the subvolume mode is. So to start, I'll click display seeds as I normally would, and you'll notice that there's a good scattering of seeds, but we're going to need to place a lot more because they're not on the prominent branches in our region of interest or our subvolume. So I'm going to raise this up to something like 90, which is a little bit egregious, but for the demonstration, I think this will work well. And then I'm going to change the density. Because the seeds are placed so far apart, I'm going to change this to dense and allow the program to place more seeds in a more densely packed region. So from here, I'll click Refine Seeds and I'll validate, excuse me, and we'll be able to have the program run its sort of self-check and remove the more erroneous or egregious seeds that might be placed. I'm going to keep the seed refine filter at 3. I apologize. I'm going to keep the refine filter at 3. And you'll notice that right now it's only on these major branches, but that's okay. Although the seeds are placed here, there is enough saturation on most of the larger branches where the program will be able to pick it up. Again, I'm going to keep the sensitivity. I might lower it just a little bit to 65 rather than keeping it at the default. And then I'm going to click Trace. We'll allow the program to run through and create the neuronal structure here. And you'll notice that it did a fairly good job. And for demonstrative purposes, I'm not going to go through the editing process here, but the editing within the subvolume mode is the same as it were outside. So we'll say that this region is good. And I'm going to return back to, once again, view the full image. And you'll notice that the data has remained. The data that we've created, although we're going to create a new subset or another stack, the data will stay as it is. And we can go and now change this subvolume to acquire a little bit larger of an area. And when I click View Subvolume here, you'll notice that the tracing above is present, but we might want to just focus on what's below here. So what I can do, rather than having to shift the entire image, I can click this clip button. So it's between our show and hide tracing and the snapshot. And what it's going to do is it's going to hide the tracing that was above the region, or anything outside of our current subvolume. It's going to hide those, and we can always bring those back. And so you'll notice here this nice orange branch that's been made. When I click the clip option to hide the tracings, it will remain because it's actually in our current section. So if you see some sort of fragments, that's why that is why they're present. It's because they have made their way into our current subvolume. Again, because the somas here are, there's a lot of them, rather than taking up a lot of time to kind of allow the program to automatically detect these somas, what I'm going to do is I'm going to detect a couple of them manually, and then I'm going to go through and use the automatic tree tracing mode. And we can see that the settings that we use here are going to be vastly different than what we used in the above subvolume region. And so we're kind of pushing again the fact that you don't need to keep the same criteria for both of your subvolumes. So again, we'll show the seeds, and then I'm going to lower this down to 70. This is typically the default setting, and this is fine for now, but I'm going to change this back to medium. We no longer need the seed density set to dense because we have such a dense network below. The seeds are being placed enough. Maybe I can raise this to 80. And then when we go to the refined seeds, I'm going to validate, and we'll see again the seeds being removed. The next thing that I'm going to do is I'm going to remove the seeds that are on some of the somas because we don't need that information being traced. Let's. Nope, I clicked something by accident. I apologize there. So let's just return, and then we'll click trace, and then we'll do the directional kernels method. So there's a question on here. Let's see. I'm not sure quite what happened there. Let's return to the full image and we'll see. Somehow our subvolume got clipped there. So I'm going to return back and then I will answer that question. I apologize. So let's return, view subvolume. We'll go to tree and we'll try that again. Okay. When you create a new subvolume, does it not continue the tracing into the new subvolume? When you create a new subvolume, does it not continue the tracing? When I create a new subvolume, no, it does not continue the tracing unless you're working in the user guided mode or the smart manual mode. With the automatic mode, what we're doing is we're limiting the searching algorithm for the branches or the puncta or the spines. Any mode that you're in where you're doing an automatic detection, we're going to be limiting the search region to that specific subvolume. When you're working with something that's more manual or the user guided mode in the tree panel, for instance, it's going to allow you to continue tracing, but it won't do the tracing for you. And that's typical of the user guided mode. It does need that user intervention. But to answer that question, there's not going to be an automatic process to move throughout all of the subvolumes. But what you can do is once you've finished your branch tracing, I can always return to the full subvolume, and then I can run my spine detection or my varicosity or puncta detection throughout the whole image rather than working in that small subvolume. It's nice because it does allow you to have that flexibility of keeping the different settings and regionalizing them. So in the above region, where the somas were a little less where they were a little less saturated in staining, we can always raise the sensitivity for that soma detection. And vice versa, when we're in a more heavily saturated area, we can lower that sensitivity. But I guess to directly answer the question, when you're working in a subvolume and you continue on to a separate subvolume, the algorithm is not going to automatically do that for you. It is going to require you to click and go through. Let me get back to this full screen for a second and get out of the subvolume mode. Okay, so here we have this. I'm going to open this in Neurolucida Explorer while I look through some of the other questions. Subvolume test data. And I'll save this as an XML. But I can't save it there because I didn't have permission. If a process. I read a question and read it in my head. I apologize. So if a process is outside the subvolume, it ends at the edge of the defined subvolume. Correct. If you're working in a subvolume and you have a process that hits the edge of your subvolume, but you know that it continues outside, any sort of automatic detection when you're in that subvolume mode is going to it's going to cut that branch or that detection short based on the subvolume that you have on the screen at that time. So with the data here, we can see that there is a gap, and that's because I didn't have the two subvolumes overlapping. If I was working in that user-guided mode, we would be able to see, potentially, if I was working on this purple branch and continuing on, we might see a connection to a following branch below. But because it was automatic, the detections are split into two different areas. So from here, we can just run a simple analysis and get a nice summary of what we've been doing. We can also, if I return back to Neurolucida 360, what I can do as well is if I know that I've used two different settings for our subvolumes, what I can do is I can go into our sets mode and I can create a set based on where I was within the subvolume. So by holding down the control key and having the trees and the somas and cell bodies option detected or checked off, I can hold down the control key and I can marquee and make a nice selection. And what you'll notice here is since I've marqueed, anything that has a continuation of something that was within that marquee box, it's going to be highlighted in the region below as well. So what I can do is I can uncheck some of the ones that were there, but you'll notice here that this branch goes all the way into our next, and that's a nice continuation of the last question that we received, if a process is outside the subvolume. Since this process, I believe this was the original orange one that was showing, because there was a branch that made its way into the second subvolume, when we ran that automatic detection, Neurolucida 360 and its algorithms noticed that there was a connection. It made that connection, and now we do have a process that breaks the boundary between the two subvolumes. So as I was saying, we can create a nice set here. We've highlighted our area that we want, and we can call this, let's call this subvolume 1. And then what we can do is we can do the same with the second subvolume below. We can call this subvolume 2. And now, because I've already saved the data file, when I push this icon to save and view in Neurolucida Explorer, it's going to once again save over the old data file, and then it's going to open that automatically in Neurolucida Explorer for us. So let me drag this in. And now what we can see is we have a sets option at the top of Neurolucida Explorer. When I click on set 1, or subvolume 1 rather, it should highlight everything that we've detected and put into that original set. So now if we want to, I can run subvolume 1, and I can do a summary of this. And I can also run subvolume 2 and get a nice summary of this. And we can compare and contrast. If we have a large image, we can compare and contrast the two different sets and what comprises them. So the subvolume mode is going to be really nice, whether you're working in a dense area like this or in the original image. If it's a very vast image stack that you're working in, you can compartmentalize and get different cells put into different sets from the same image stack, and then you can do a nice compare and contrast. So the last thing that I'll show within this Neurolucida Explorer program is the current Windows button. Anytime we have these tables showing, we can go and click this current Windows button, excuse me, and once again, it's going to save the tables as an Excel workbook. When I click save, it will show me the location that we've saved to, which is my desktop. I can click and load that Microsoft Excel file, and now we can do a nice comparative analysis between the two subvolumes that we've run through the analysis in Neurolucida Explorer. Excuse me. Another question that just came in is, while tracing a tree in the user-guided mode, can the subvolume be moved in the Z-plane too? To answer that question, I'm going to jump back to the original large data file or image stack that we had open. And we can see that in the Z-axis, this is a pretty slim Z-axis, but what we can see here is that there is variation in Z. So what I'm going to do is I'm going to just jump up to an area that we have a good sort of transition in this Z-axis for. I'm going to generate the subvolume here. And as I trace, it's going to follow the process as we go down. So we'll just load this little region. I apologize for the delay. We're also close to the time limit, so I'll answer the question as I show it. As we're tracing, when we hit the boundary, the program is going to follow what we're doing, and it should change in Z for us. So now, whoops, let me undo that. When we get to the boundary edge, it will follow in Z. And then because this wasn't at the very edge of the boundary, it didn't make its way. There we go. Now we're loading the next region. But you'll notice if I rotate here, I apologize for the confusion there. As I was going down, I made it to the edge of the boundary. And then it's actually pushed back our subvolume. So it is going to be moving that boundary in Z for us as well and having a nice continuous tracing for us. There are a lot of questions that came in that I did not get to, and I apologize for that as well. So I will, in a follow-up, I'll do my best to try and answer these specifically to the people that had asked them. But unfortunately, we are at our time for today's session. That went by a lot more quickly than I was anticipating. Thank you for my rambling. Thank you for listening to my rambling at some points. I really appreciate it. The last image that we were just looking at did come from the Allen Institute, so I did want to acknowledge and say thank you to them for allowing us to use their image data. And lastly, as I'm wrapping up, once the webinar ends, we will be sending you this recording if you'd like to your email. But once we close down, there is a short survey after. Please let me know how I did. Please let me know if there are more topics that you're interested in specifically, and how we can kind of cater these short form webinars to you and your research and what you guys are interested in. Again, thank you very much for attending. I appreciate your time as always, and I look forward to seeing you in 2 weeks. Yeah, in 2 weeks. Not this coming Friday, but the one after. Again, thank you. Enjoy the weekend, and I look forward to talking to you all very soon.